So the purpose of this activity is twofold. We want to make sure we understand how the scientific method works and we're still able to design a proper lab. We did that once in the last unit. We're going to do it again here in this unit. Okay? Things to be aware of when we're designing a lab procedure. Our procedure must be controlled. Okay? That is, we are only testing the effect of one factor on plant growth. The way we make sure of that is we take into account controlled variables. Okay? That is, we make sure that anything else other than the factor we're trying to test is being kept the same for all of our plants. All right? I will give you a prime example of how people do this wrong. Okay? They say to me, I want to do an experiment that will test the effect of the environment on a plant. How many different factors might there be in an environment?
Light.
A lot, like all of them. Okay? That doesn't tell me anything about what you know about factors that affect the growth of a plant. Okay? I can't test one plant growing in the desert and one plant growing at the North Pole and say that that's a controlled experiment. Okay? All I'll know is that plants die at the North Pole. Okay? That's not helpful. So you got to test just one thing. Okay? That one thing can be nutritive, so it could be like the amount of water or the amount of sunlight or the soil, anything like that. You pick what it is. Okay? Just make sure it's only one thing. Okay? And I've had people get pretty creative. Like they tested watering plants with different liquids. Okay? Or they tested different colors of light. Okay? As long as you're only testing one thing, the color of the light or the liquid being given, you're running a controlled experiment. Does everybody follow what I mean? Okay. All right. So what you're going to do in this experiment is you're going to first write a short abstract. Okay? Now, an abstract is something we haven't done before, okay? Because we don't put them on lab experiments. But for this one, what you're writing in the abstract is basically what you know about plant growth and how it relates to the factor you want to test. In real science, to get funding for your research, you have to sell it. Okay? That's kind of what the abstract does. It's kind of a sales pitch. Okay? So you would want to say, something about what you know about how light affects plant growth, and that, if you run this experiment, it might lead to increased productivity of agriculture or something. Okay? Like you just have to, basically it's 2 or 3 sentences where you're just telling me, here's what I know about plant growth, this factor, and what it might be used for. 3 sentences. Okay? Super quick. After that, it follows the same format as all of our labs. Problem. Okay? So you've got to come up with a problem. What is the effect of light on plant growth? Whatever you're choosing. Okay? Or which color of light works best for plant growth? You decide. That's your problem. Literally. Both ways. It's what you're going to write, and it's your problem. You have to do it. Okay. Then you've got your design, same as always. What's your manipulated variable? Well, it's the factor you're testing, so talk about it. How are you manipulating it and why? Okay? What's your responding variable? What is everybody's responding variable here? Plant growth. Okay, now, hold on. Before you just go writing plant growth. Okay? There are a lot of different ways to measure that, and it could be dependent on what plant you choose to use for your experiment. Okay? So think about how you might best be able to measure the growth of the plant you're going to choose to run your experiment on. You might want to just take pictures of it, or you might want to record the mass of the entire setup over the course of the entire thing, or you might want to measure its height, or count the number of leaves. There's all kinds of different ways to measure the growth of a plant, and you may want to have more than one way, okay, in order to have valid data about plant growth in your experiment. So when you say responding variable is plant growth, as measured by, and talk about it. That's your how and your why. Okay? Everybody with me there? All right. 3 controlled variables. Think about the other things that you need to make sure you keep the same so they don't affect the growth of the plant. Okay, otherwise all you're doing is testing more than one thing. Okay, we only want to test one thing here, so you have to control the other stuff, okay, that would affect the growth of a plant. Okay, explain how each controlled variable is being controlled and why each controlled variable. You don't just write, it would affect the growth of a plant. Tell me how. Okay, obviously it would affect the growth of a plant. Tell me how. Okay, then you'll have your hypothesis. If insert factor here affects the growth of a plant, okay, and describe your experiment briefly, then predict your results, okay, whatever you think they'll be. If you're doing this on a flowering plant, maybe you're going to predict how many flowers it's going to have, okay, or you know, these plants will grow more flowers, or this plant will produce more fruit, whatever, okay, you predict your results. Then you're going to have your list of materials, okay. This doesn't have to be rocket science. What do you need to grow a plant, okay, and list it. But also consider listing the kind of amounts of things. I mean, you can put, like in your materials, soil, pots, whatever. But in your procedure, you need to tell people how much soil, how big a pot, okay, things like that. Everybody with me?
What's the standard amount of soil?
Well, it depends on what plant you're using, okay? That's, I mean, in the end, the plants we used yesterday, okay, that's a 2-liter pot. Okay? I mean, unless you're growing oak trees and doing this experiment over 3 generations, right? You probably don't need more than, you know, maybe 4 liters of soil per plant. I mean, again, it depends on what size. If you're just growing a plant like this, then you only need 2 liters in a 2-liter pot. Okay? Also, keep in mind how much water you're going to be giving the plant. I've had people say, and water the plant with a cup of water a day. That's a lot of water every day. Okay, I know, like a cup of water to us, like, yeah, we got to drink like 2 liters of water in a day. But a cup of water a day for a plant will kill it. Okay, it's a lot. It'll just be soppy and leaking all over the place all the time. So be mindful of how much water you're giving it, especially if water is going to be your manipulated variable. Okay, you don't want them all to have the same result, which is they all drown. Okay, so just kind of keep that in mind. All right. Now, incidentally, when you're setting this up, you do need to have one plant that's kind of your control. Okay, your comparison plant. So if I'm doing an experiment on the amount of water a plant gets, I need to have one plant that gets how much water? Right. Because I need to know what happens to a plant that doesn't get any water. Obviously, common sense says what will happen to that plant, but I scientifically have to prove it. Okay? I have to have a comparison. Okay? Plant with no water, crunchy. Okay? Plant with 2 milliliters of water per day, crunchy. Okay? Plant, you get the idea, right? Like you have to have a comparison plant, right? If I'm testing light, I probably need to have a plant that's in the dark. Okay? As a comparison, as a control group. Okay? So in your procedure, make sure you're telling them in detail how to set this up. This is where I check that the controlled variables you listed are actually being controlled, because this is where you tell me how you manipulate your manipulated variable, how you measure your responding variable, and how you control your controlled variables. Okay? This is where the detail comes in about how that happens. Okay? Every plant is growing in a 2-liter pot with 2 liters of potting soil I bought at the garden center at Wally World. Okay? Whatever. Okay? You gotta tell me how am I controlling this one. Okay? And so on and so on. All right? In your procedure, you also need to, in detail, tell me how you're measuring the growth of the plant. Okay? And how you're manipulating the manipulated variable. Okay? That's it. It ends at procedure. We're not running this experiment. It's imaginary. Okay? It's what Newton used to do a lot of. Thought experiments. Okay? When you get to the point Newton was at, you didn't even have to do real science anymore. You just thought stuff up and people went, okay. Right? So we're at that, we're going to imagine we're at that point. We're just going to design this experiment, but we're not going to run it. That does give you a bit of freedom in that it can be a little less practical. Okay? I mean, you could do this on an oak tree and say, my grandchildren are going to finish this experiment, okay, 100 years from now. Right? That'd be fun. Okay? All right, questions on what you're doing? All right, then get to okay.
Oh, it was just about the abstract.
Okay.
So it's just like
what you know about plant growth, what you know about the factor that you're choosing, and some practical application.
Okay.
3 cents.